Hello, everyone. I'm Christy Risk, Senior Editor at GenomeWeb, and I'll be your moderator for today. The title of today's webinar is Reconstructing the Transcriptomic Vector Field from Metabolic-Labeling-Based Single-Cell RNA-Seq Data, and our sponsor is Lexogen. Our panelist today is Dr. Xiaojie Qiu, postdoctoral scholar of cellular molecular pharmacology at the University of California, San Francisco School of Medicine. You may type in a question at any time during the webinar. You can do this through the Q&A panel, which appears on the right side of the webinar screen. And if you look to the tray at the bottom of your window, you'll see a series of widgets to enhance your webinar experience. And with that, I'll turn it over to Dr. Qiu. Please go ahead.
All right, thanks for the nice introduction. It's my great pleasure to talk here. So the topic that I'm going to talk about is mapping the vector field of single cells. I think some of you may already saw my preprint on this last year. We actually also have a software toolkit, so you can download and play with it. One of the fundamental questions in biology is that how can a single cycle be able to differentiate into a variety of different cell types? We know that during this process, the hematopoietic stem cell can choose different trajectories to become either the myeloid or lymphoid lineages. And those progenitor cells can further choose a different trajectory to become either basophil, erythroid, or other types of terminal cell types. In order to study this dynamic process, in the past, people tend to use some time series approach to measure the transcriptome of the cell at a different time point. So with this kind of approach, you can measure the average expression of a mixed population of cells at a different stage. The past has already able to show we are able to identify the marker genes associated with each stage. However, because we are looking at the mixed population of cells, we are not able to see the detailed dynamics of each individual cells. Recently, in the past few years, the emergence of a single-cell RNA-Seq provided us the capability to look at each individual cells. So with some algorithms we developed, then we can automatically learn the trajectory that represent the cell progression. This kind of operation is called the simultaneous. And the basic idea is that we can orient the cell based on the similarity of the single-cell transcriptome. The simultaneous ordering was turned out to be very powerful and popular in the past few years. They can tell us the key transcriptional factors that they dynamically change or bifurcate between alternative lineages. However, it also has a few limitations. Firstly, the simultaneous only tells you the central trend of cell-fate transition. Secondly, for the majority of the tools, you need to tell where is the root of the differentiation process. Meanwhile, there is another very fascinating development in the single-cell field. Namona and others find that although the majority of single-cell platform tries to enrich for mature mRNA, they can still get around 20% of the reads from the introns. And then they can use a classical gene regulation model to study the derivative of the gene expression state. They call this RNA velocity. Basically, RNA velocity represents the first derivative of the spliced mRNA over time, or beta U minus gamma S. Here, beta is the splicing rate, gamma is the degradation rate. Well, U and S are the unspliced and the spliced mRNA. In order to make this system to be identifiable, then they assume a universal constant for beta across all the gene. So beta equal to 1. And then by looking at the phase portrait of spliced and unspliced, they can treat the cells at the extreme point as the cell at the steady state, and then they can perform a linear regression for this for those extreme cells. The slope of this linear regression is essentially the parameter gamma. Once we get the gamma, then we can estimate the velocity for each individual gene in each individual cell. So respect to each cell, the RNA velocity is a high-dimensional vector. We need to project this high-dimensional vector to low dimension. And then after the projection, we can then perform some local averaging and smoothing. And then we can use the grid of query plots or string length, or even name convolution, name integral convolution, or other tables of vector field visualization tools. So RNA velocity was very well appreciated in the community. That has been used across many different data sets or different systems. However, it has a few limitations. Firstly, it only tells you the short-term direction of magnitude of RNA dynamics. Secondly, for the most part, RNA velocity is just used as a descriptive instead of a predictive tool. Lastly, because they have to assume a universal constant for beta, so the parameters are relative, so it doesn't really have any physical meaning. Here I would like to mention that there are also a lot of very exciting progress in this direction. The excellent tool from the SCVelo tool from Fabian's thesis group tries to extend RNA velocity to non-steady state, and the protein acceleration work from the Leo Paglis lab also generates RNA velocity to protein velocity. However, for today's talk, I would like to shift the emphasis and tell you that we can go beyond the RNA velocity to learn the vector field. So what is a vector field? And what can we do with it? To simply put, the vector field can be represented as a vector valued function that map any point in the gene expression space to another space. So it can include the velocity, acceleration, or other types of vector field. Shown here, this equation represents a velocity vector field. So what can we do with it? Since we can use it to map the full trajectory of a cell field on the entire gene expression space. So assume we know the vector field of a system shown here, and then we can pick any point on this space and take it, treat it as the initial state, and then we can release this cell on this vector field. Then the cell will travel along the vector field, following the velocity vector at each point on this field. Then they will eventually convert it into a terminal cell type. And the trajectory it passes through in this space is the path the cell will evolve in the gene expression space. And the vector field can also be decomposed into two different components: the core-free component, which can be used to study the differentiation process, and then we also have the divergent-free part, which can be used to study the cell cycle. You may didn't notice this, but actually vector field is everywhere in our normal life. So it needs to be studied for the airplane flight, and also be widely studied in different sports, and of course in magnetics and even the worldwide climate changes. So under the context of vector field, we can then think of the cell differentiation as a traversal in the gene expression space. We know that the cell state can be represented as a vector X, which consists of the concentration of different genes like S1, S2, and S3. Then the interaction between these genes forms a network. This network can be formulated as a function F. This function F determines the progression of the cell in the gene expression space. So shown here, we have three states, S1, three cells, S1, S2, and S3. So just represent their current cell state. But if we know this regulatory network, we can actually predict the historical and the future cell state. And on the other hand, we also notice that no matter where the cell located initially, they eventually converge into a terminal a steady state, the A1 state. In fact, the regulatory network I just mentioned corresponding to the vector field function F. Of course, the topic of today's talk is to tell you how can we actually starting from samples of ion velocity to get a vector field function that can be applied on the entire gene expression space. But let's assume we can get that, then we are actually in good shape, because there are more than 100 years of research on this field. So we can then take methods from dynamic system to estimate the steady state of the vector field. The steady state can include different types. Normally, we can classify the steady state into stable steady states or attractors. Or we can also identify the non-steady states or just saddle points. Here, we can also identify some characteristic plane or lines that can be able to separate the boundaries of the vector field. So, for example, the separatrix defines the boundary between different domains of attraction or attractor basin. So in this system shown here, we have 3 attractor basins, B1, B2, and B3. And all the cell state belong to each attractor basin will eventually attract to a particular attractor state. Just attractor state are like a black holes. So my idea to treat a cell as traversal in the high-dimensional space actually doesn't come from nowhere. It was actually deeply inspired by a few papers I read a long time ago. As early as 2005, Xiaojie and colleagues first demonstrated that the cell fate can be treated as attractor states of a high-dimensional gene regulatory network. In this study, they take 2 different drugs, DMSO and ATI, to stimulate the HL60 cells, just as the cancer cell for the progenitor HSCs. And then they performed a 10 series of the transcriptome of user cells during this differentiation using microarray. They find that although initially the cells departure from each other because of the difference in the stimulation, but they eventually converge into the same destination, because the final neutrophil state is the attractor state. Of course, this kind of research was done almost 20 years ago. Nowadays, we can definitely do much better. But what is the best way to measure the dynamic cell state? Recently, in a paper published with how much money, we show that the best way to measure the dynamic cell state is using some metabolic labeling based approach, like the single cell nascent, scaffold, SCNT, and so on. The reason is quite simple, because when you use just metabolic labeling based single cell assays, you are actually measuring 2 modalities of the same cell. You can look because the metabolic labeling can tell you which mRNA is out, which are new. Then the question is, now let's talk about how can we actually quantify the nascent RNA with metabolic labeling. Overall, there are 2 different methods, the biotin purification-based approach and the chemical conversion-based approach. The beginning of these two approaches are quite similar. Both requires using different labeling strategies to label the cells, and we normally use the FSU. And then for the biotin purification, you need to use thiol-specific biotinylation to add some tags to the labeled mRNA. And then you can use the streptocopy beads to pull down and separate the newly synthesized RNA from the old RNA. For the chemical conversion, you actually don't need to separate the new RNA from old RNA and prepare two different libraries. So what you do is you perform a chemical conversion, either using the AAA alkylation or using some chemical, the hydrogen bond reconfiguration with the 10 levels Seq protocol, or use the TUC-Seq to artificially create a T2C mutation. And in the end, we can then identify the reads that have high confidence of T2C mutation, and those reads are corresponding to the new RNA. So you can see, compared to purification approach, the chemical conversion don't need to avoid the problem of cross-combination and also the normalization from the purification approach, because they need to separate the new RNA and old RNA into two separate library for sequencing. And the Lexogen actually provide a very nice solution for doing this, for doing the SNN-Seq. And at the beginning of my project, I used this product. It works quite easily in our hands. Also, you may notice quite a lot high-profile paper which use this technology. But of course, this method is bulk based RNA-Seq. I believe they may also work on some single-cell versions. So stay tuned. Okay, I just mentioned we can use metabolic labeling for doing, mostly talking about the FSU. But the metabolic labeling strategy actually was widely adapted for the past few decades. So there are many other options. So you can use BRU, EU, or biotin NTPs. And for the FSU, there are quite a lot of different bulk RNA-Seq approaches. There are SNN-Seq, TUC-Seq, TT-Seq, or 10-level-Seq. And recently, a few groups adapted the SNN-Seq chemistry to plate-based single-cell RNA-Seq. So the SC SNN-Seq nascent-Seq was developed. And the scale-fed approach was developed around this combinatory indexing approach. And it also uses the SNN-Seq chemistry. But the next thing about the scale-fed is that you can produce easily thousands of cells or even millions of cells. And then I will talk a little bit more on the SC&T-Seq next. For the another thing I want to highlight in this slide is that recently Alexander von Alden and the group adapted the EU-based labeling strategy to develop the SC-EU-Seq. This is a purification-based approach. I was very fortunate to meet two postdocs, Qiu and Pen from Hall's lab at the University of Pennsylvania late last year. Since then, we started to collaborate. And then Qiu and Pen recently developed the SC and T-Seq method. This is the first drop-seq-based approach for doing the metabolic labeling. So they can produce thousands of cells quite easily. And if you compare SC&T with other methods, you can see with relatively few number of reads per cell, you can get quite a high number of genes and quite a high number of almost like 17,000 UMI per cell. So overall, SC&T-Seq, especially the second string synthesis approach, is preferable. Okay, so we can actually be very creative and clever in design different metabolic labeling strategies. So, for example, you can either design the expansion where you take different days, and at each different day you perform different you label the cell for different period of time, and then you harvest the cell. We call this is a multi-10 series experiment. And this kind of experiment can help us to estimate the transcription, splicing, and degradation parameters alpha, beta, and gamma. Similarly, for the kinetics, you can also design a kinetics experiment. For the kinetics experiment, you can choose a typical time point and label the cell for different period of time and the harvest then. For this kind of experiment, you can also try to estimate the parameters alpha, beta, gamma. But as I will tell you later, it's optimal to estimate the transcription rate alpha. And for the degradation, you first label the cell for extended period to saturate the labeled RNA, and then you can wash out the FSU replaced with excess normal U, and then you check the cell after that for a certain period, which is a kind of experiment. It's optimal to estimate the splicing and the degradation rate. And then you can combine the degradation approach with one-shot experiment so that you can estimate the alpha at each day, for example. And then you can also, you don't need to label multiple time. You only need to label one time point for each day. What I'm just showing here are typical metabolic labeling strategies, but you can also think of more complicated cases where you could combine the canonical with the experiment with the degradation experiment to get a mixture type of labeling strategies. So in order to deal with all these kind of labeling strategies, we developed a very comprehensive model in Dynamo to handle this. Our model dramatically expand the original RNA velocity framework. So we need to first consider the switching between the promoter states. So the active state can switch into inactive state and vice versa. And then our model need to explicitly consider the metabolic labeling. So this can lead us to have full species UL, UUSL, SU, or unspliced labeled RNA, unspliced unlabeled RNA, spliced labeled RNA, and spliced unlabeled RNA. After the splicing, the spliced RNA can also further go through the translation step to become the protein, and then the spliced and the protein can degrade. So the incorporation of the protein can help us to study the set-seq or rep-seq or other types of protein RNA co-assays. This is our most comprehensive model. However, for different types of experiment and for different types of data, we need to make some we need to make some reductions for this comprehensive model. For example, if we are dealing with regular 10x data, then we don't have labeling. Our model need to handle that. And then for some time, for some cases, you don't need to split your labeled and unlabeled RNA into spliced and unspliced RNA because if your labeling strategy, your method is not that sensitive, splitting into full species, will make your expression very low, and it says you can't really estimate anything. So for this kind of scenario, we also need to deal with that. So then essentially we build a universe of estimation framework in our tool. So we this framework basically has uses two different assumptions: the steady state and the canonical assumption without assuming steady state. And for the steady state assumption, we have three different models: the linear regression, generalized method of moments, or a negative binomial distribution-based approach. So the first approach is similar to the RNA velocity, and the second one is quite similar to the SCVL's moments approach. But of course, we made some. We also extend this approach that to explicitly deal with the metabolic labeling and also different types of labeling strategy, including kinetics, degradation, and one-shot. And for the negative binomial distribution, we can use it to study the RNA boosting. And on the other hand, for the kinetic assumption, we need to use two different models, the deterministic and also stochastic model. For the deterministic model, we can use it to deal with a kinetic one-shot and degradation experiment. And we also need to deal with different data types, like if your data has splicing or no splicing, or if your data has labeling or low labeling. On the other hand, for the stochastic model, we can use it to deal with the kinetic or degradation experiment. And for kinetic degradation experiment, we can also need to deal with different model assumptions and a data type. We can consider our model include the promoter switch or without the promoter switch, include the splicing or no splicing, include labeling or low labeling. So this is very sophisticated and very comprehensive. We essentially consider almost every type of modeling strategies. But for the sake of 10, here I'm going to just use the canonical assumption and the dimensional model to explain how can we use it to estimate the parameters for the canonical experiment. And we assume we have full species, spliced and unspliced. So because we are using a deterministic model, so we don't need to get into the chemical master equations. We essentially just look at the ODE for the unspliced and the spliced mRNA. Because Jews are first order linear ODE, then we can solve it to get the analytical solutions. And then once we can take those equations to feed the 10 series of the labeling data, and then we can estimate the parameters for alpha, beta, and gamma. So here I'm gonna demonstrate using the kinetics data from the SCEU-Seq paper. So for the kinetics experiment, we know that the unlabeled RNA will in general always decay, because for the unspliced unlabeled RNA there is no inflow, so it always spliced into spliced unlabeled RNA. So it will always decay. But for the spliced unlabeled RNA, if the degradation rate gamma is larger than the splicing rate, which is often the case, then the spliced unlabeled RNA will always also decay. On the other hand, for the unspliced, for the labeled species, it will always increase until it saturates. And on the 10th scale of a kinetic experiment, it normally won't saturate. And so that's so the unspliced labeled RNA and spliced labeled RNA will always increase for a kinetic experiment. So then we take the 10 series of data of the unspliced unlabeled RNA and assume the first order decay model to estimate the parameters beta. Once we get the parameter beta, we can then, using the analytical solution of the spliced RNA to to feed the 10 series of the spliced unlabeled RNA and to get the parameter gamma. And similarly, we can also take the estimated parameter beta to feed the 10 series of unspliced unlabeled RNA to get the parameter alpha. What I just said here tries to estimate each of the parameters using different set of data, but in fact, what we do in practice is to use all those data jointly so we can get a global optimal value for each of the parameters. Here I'm going to show you show you some results when we apply this approach to the kinetics experiment for the SCEU-Seq data. So the experiment design for this data set is actually quite smart. So they use two fluorescence, GFP and IFP, to stage each cell. And orthogonally, they also try to perform a kinetics experiment. So you can essentially take different pieces from this cell cycle progression trajectory to get a kinetics experiment for that stage, and then you can take calculate the time-dependent or stage-dependent alpha, beta, gamma across the cell cycle. Here we just take a small piece from this stage, and we just highlight the fitting for one gene. You can see in general our fitting works quite well. Also, you can see the labeled RNA is expected, they always increase, whereas the unlabeled RNA also expected it's gradually decrease. However, we do see that the unspliced unlabeled RNA in general has very low expression. This indicates that for the kinetics experiment, it's not ideal to use unspliced unlabeled RNA to estimate the beta and gamma, but it's good. It's optimal to use the labeled RNA to estimate alpha. All right, let's also talk about how can we use the degradation data from the SCEU-Seq data for SCEU-Seq paper to demonstrate how can we use it to estimate the parameters for gamma. So for the degradation data, we can look at the labeling RNA for the look at the fitting for the labeling RNA. So for in the degradation experiment, because we force to saturate the labeling of RNA and then wash out, so the label RNA will always decay, and we see that the two genes which are ESC activity genes showing showing at the beginning has a much shorter half-life. It's nearly just 2 hours. While the GABA DH, which is a housekeeping gene, the half-life calculated based on the gamma fitting is much longer. This makes sense because for the activity gene, you want them to have a much quicker turnover, but this is not required for the housekeeping gene. On the other hand, if we look at the fitting for the unlabeled RNA, which was used to estimate the alpha, you can see the fitting doesn't work very well. This also makes sense because the fitting of unlabeled RNA tries to get the transcription rate. However, the transcription rate change on the scale of minutes, but the experiment we done here lasts for more than 24 hours. So we again just demonstrate for the degradation experiment, it's optimal to estimate the gamma, but not optimal to estimate the alpha. So in fact, we provide a guideline to tell you what kind of parameters we should estimate from each type of metabolic labeling data. We incorporate all those estimation framework into our toolkit, and we provide a easy workflow. You can follow it, and also we provide 8 different tutorials, including two from metabolic labeling based single cell assays, the SNN-seq and the nascent-seq assays, to show you how can we apply our tool to analyze different data. We are also planning to release more notebooks related to the SCEU and the scaffold data sets in a couple of days. Okay, but for now, let's try to demonstrate how can we apply Dynamo on the labeling data to understand the neuron activity response. So this is a collaboration with Wu's lab. So recently, Qi and Pan harvest the cell from the mouse cortex at embryonic day 16 and dissociate this cell into a media, and then they label the cell with full SU for 2 hours. But they also added the KCL at a different time point during the labeling. and then the harvest cell for SCNT-seq sequence, SCNT-seq. So we then applied the Dynamo to estimate to the labeling data to obtain the time resolved RNA velocity, shown on the left. We can see that the time resolved velocity shows two distinct stages. So for the first stage, the cells at 10.0 will rapidly move toward the cells at 15 minutes. And for the second stage, the cell can go in backward, but they still follow in the time progression from 15 minutes to 30 to 60 to 120. And we also see that the first stage has very strong velocity indicated by the thickness of the strand. However, when we look at the splicing velocity data, just based on the spacing and spliced and unspliced reads, we are not able to see this nice this progression, and the velocity in general is very messy. So when we look at the phase portrait of a few earlier and late response gene, neuron activity gene, we can see the EGR1, FOS2, those are two earlier neuron activity gene. So the velocity of this gene turn on much earlier compared to the late response gene, hormone 1. In comparison, if we look at the phase diagram for the splicing data, we unfortunately we can't see almost no expression for the EGR1 FOS and FOS. This is probably because these two genes doesn't have AT enriched intron regions, and it also explains why the velocity flow looks really messy. All right, so now I talked about how can we use our framework to estimate the good parameters for alpha, beta, gamma, and how to resolve how to obtain the time resolved RNA velocity on the labeling data. Next, I would like to show you how can we move from RNA velocity to get the vector field function. So after the RNA velocity analysis, we can obtain the samples of velocity at the different cells. And some of these cells may be incoherent to the velocity for these cells incoherent to other cells nearby. Then we have a procedure to identify these outlier cells. Once we identify these outliers, we can then apply a method called the sparse vector field reconstruction to learn the vector field function f. And then we can apply this vector field function to any point in this space, even on the places where we don't sample the cells. Also, our vector field reconstruction method is very efficient. We actually can work on thousands of dimensions and also 10,000s of cells. After the vector field reconstruction, we can then try to project we can try to characterize the vector field. So the idea is so I'm not going to too much details, but the idea is that we wanted to expand the vector field into a set of basis functions. So here for these three basis functions, the velocity vector flow in each of them are very simple. But when we combine them together, we can see it can be used to represent the complicated vector field. So how does it look like in real data? In practice. So first, perform a simulation, and then we take some samples from it, and then we reconstruct the vector field with Dynamo. You can see the vector field we reconstruct is nearly indistinguishable from the true vector field. We can also apply this to more complex systems and get similar results. And then we wanted to validate this on the real data sets. We take the Larry data sets from Alan Klein's group, in which they use the studied barcode to label the progenitor of the hematopoietic stem cells, and then they sequentially sample the cell multiple rounds and using the in-drop-seq. We believe this is a good data set for validating our vector field. So I take that data and reanalyze to get the spliced and unspliced accounts, and then perform the RNA velocity analysis with Dynamo. So then we can see very nicely the undifferentiated cell can choose two different trajectories to become either the neutrophil or monocyte lineage. And then I also developed a method that tries to automatically summarize the cell-fate transition to build the models of cell type transitions. So then we take just samples of RNA velocity and learn the vector field function on the entire space. And then once we get the vector field function, we can then calculate the fixed point. So in this system, showing here, we have three different types of fixed point, including the empty cycles, which correspond to the repressor, or the just may correspond to the unstable progenitors. And then the half-field cycles, just saddle points, just also corresponding to the progenitors. And then the field cycles, just a stable fixed point. Indeed, we see near the tip of the neutrophil or monocyte lineage, we can identify two field cycles indicating just a cell at the steady state. And we also find a few half-field cells near the undifferentiated region, just may indicate just unstable states. We then also try to calculate the lower colline for this system, and indeed, in the section between this lower colline corresponding to the fixed point, there are actually a lot of things to be explored for this data set. But for the sake of time, I'm going to take last few minutes to tell you how can we move vector field to potential landscape. So potential landscape is one of the is very well appreciated and intuitive the concept that was used in many different fields in biology, including the epigenetic landscape from Conrad Waddington, used in development, and the energy landscape in the protein folding, the fitness landscape in the evolution. So after we get the vector field function, then I used a method that was developed almost 20 years ago to map the potential landscape. Without going to the technical detail, let's look at how does it perform in practice. So here I'm taking the hematopoiesis data set from the original RNA velocity paper. Also reconstruct the vector field for this system. So you can see for the astrocyte, OPC, granule, CS3, just the full states are corresponding to stable cells states. Where for the neuroblast state, just the cells looks like at the progenitor state, so they are certain points. So then we applied the potential landscape mapping method on this data set, and obtained this picture. So the y-axis corresponding to the probability of the cell at that particular state. So you can see for the terminal cell type, CS3, granule, astrocyte, OPC, the probability is very high. However, for the neuroblast cell, the probability is quite low. I was actually really excited about this result because this method was developed almost 20 years ago, purely from the perspective, the theoretical perspective. But now we can actually apply it to study the real, to apply to the real data set. All right, and I think, and hope I convinced you that the Dynamo is a comprehensive framework that can be used to model the RNA dynamics, and it can be used to study different types of modern labeling strategy, including kinetic degradation, one-shot, and other types of mixture labeling strategies. And secondly, I hope also convince you that Dynamo is the first tool that perform correct velocity analysis that explicitly models the labeling and also consider the labeling time. Thirdly, I hope I will Also, the Dynamo can help us to reconstruct the vector field in the full transcriptome space. And lastly, Dynamo is able to quantify the global cell state by mapping the potential landscape. With that, I'd like to thank Jonathan for helping to organize support for this project, and also Joseph, Angie, Dion, and many others from the Boltzmann lab who helped me doing a lot of experiments. And I would also like to especially thank Yan from Xin's lab, who helped me to develop a lot of series for this project. Without his help, this is not this project is impossible. And also, I would like to thank Xiaojie from the Dynamo lab at Bellhub, who helped me doing a lot of single-cell forensic library preparation. We actually have a really nice experiment on that, but for the sake of time, I'm not able to talk about it. And also for my fantastic collaborator, Qi Pan from House Lab, we had a lot of fun to discuss the single cells NT-seq and also analyze the metabolic labeling data. And also my previous collaborator, Lee, Luke, Alex, Sarah, Emma, and many others who helped me to discuss how to reconstruct the vector field and mapping the potential landscape. All right, that's all I have today. Thanks for your attention. I'd like to take any questions.
All right, thank you, Dr. Qiu. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. Before we begin the Q&A, we'd like to ask attendees to take a moment after the webinar has ended to give us your feedback by taking our exit survey. All right, we'll now start the Q&A. So our first question, is it possible to establish standard RNA velocity values for specific contexts, such as healthy cells in different stages of cell cycles, for cancerous cells, for recently infected cells? And if it is possible to do that, is it meaningful to do it?
I think it's definitely, you definitely can do that. And if in order to do that, you probably need to do the RNA velocity for different groups of cells. If, for example, if you have some samples include healthy cells or cancer cells, you need to separate these two groups, either using some clustering or other method to separate them, and then you can perform RNA velocity for each of them. And then you can also do some comparison between them to see whether the RNA velocity or vector field changed between these two different states.
And how do you determine the rates of transcription and degradation of mRNA in the cell for vector field and trajectory analysis?
As I mentioned, we are actually taking full advantage of the metabolic labeling data to estimate the transcription, degradation, and the splicing rate. The basic idea is that for the labeling data, you can controllably to measure the nascent RNA produced after a time period, and then we can use the ODE or other master equation to model the time series of just different species to obtain the parameters for the transcription, degradation, and the splicing rate. One thing I wanted to mention is that doing this for the regular 10x data or other tables of regular single-cell RNA-seq data is difficult because the splicing, because we are just measuring the splicing. So since just splicing are intrinsic, so there is no way we will know how long the splicing process has happened. So that's why we use the metabolic labeling approach to explicitly model the time and to get the parameters with physical meanings.
And how does the metabolic state correspond to the transcription program and dynamics?
Sorry, what do you mean stage?
The metabolic state. There was a a slide, I believe, where you were speaking about the metabolic state. We can move on also if you don't have an answer to that.
I think if I understand the question correctly, the metabolic labeling actually doesn't affect the cell at all. So I would say it's just a way to help you to measure the nascent RNA and this won't affect your cell. The cell can behave normally, and so I would say it's a normal state.
Okay, great. How can transcriptomic vector profiling be applied to the identification of genetic disorders or to detect mutations?
All right. Okay. So this is not really my field, but I guess to use the RNA-Seq or other transcriptome approach to identify the disease state, you need to probably perform some differentiation analysis to compare between normal cells and the disease cell, and if you identify genes that dramatically differ between these states, then maybe those genes are responsible for the disorder. But in terms of mutation, I guess it's probably difficult to use RNA-Seq to match the mutation. As a better way, you want to do is use a DNA-Seq, maybe single-cell DNA-Seq to identify the mutation during the cancer progression or other disease processes.
Can you estimate how many cells are needed to reconstruct the view of the vector field? And then is there a method to tell if this estimate is accurate?
I guess this is. I guess there is no really some rule for sum for determining the number of cells. The better suggestion is if you can generate a lot of cells, just go for it. But if you don't really have a lot of cells, you need to be careful to interpret your result. I guess this is also applicable to the analysis of RNA velocity. So if, for example, if you're doing just use the regular plate-based single-cell RNA-Seq, you only have maybe a few hundred cells. Then for this kind of analysis, you need to, first of all, to remove the cells that they are potentially dead cells or doublets. And then you also need to carefully select the genes you want to perform the RNA velocity and the vector field analysis. And then you also need to use some pre-knowledge to see whether the vector field flow actually makes sense. If you do see those, all these criteria satisfies, maybe the vector field you learned is correct. But in general, I think a few hundred cells is on the lower end. It's better to get thousands of cells if possible.
In terms of the sequencing, can you tell us what were your shortest and your longest reads and then your average read? And also, did you do any analysis afterwards on the length reductions through the velocity path?
Okay, so we are actually using, I didn't talk about our data in this presentation, but we are actually using the single-cell SNAP-seq protocol. So the read length we use is, I think, it's 35 base pairs. I guess to quantify to accurately quantify the mutation, it's probably you do need to have longer reads. And on the other hand, it's better to use methods like scale-fed or SCNT, because those methods use UMI, and so you can collect multiple reads together to get a confident measurements of the mutation. But I guess in general you need longer reads, and also it's better to use the UMI-based approach. If the question was asking about how do we accurately estimate the mutation.
Can you tell us how mapping of vector fields could be used to determine chemical modifications in RNA?
Mapping. Sorry, I didn't really get this answer, this question.
Let me try to ask it again, but we can move on if you feel more comfortable.
We can.
Okay. So let me ask another one. Can we do epitranscriptomic studies using your technique?
Epitranscriptomic. I don't think you can do that directly because this model we are using is basically modeling the RNA dynamics. We probably could include the epigenetics in our model, but at this moment we haven't incorporated that yet.
Okay, great. Can RNA velocity over a time series be a replacement for pseudo-time ordering, or do they provide different types of information?
I think the time series for RNA velocity analysis is definitely much better than the pseudo-time approach. But on the other hand, there are some places where the pseudo-time could be easier and could be more directly applicable. Because in pseudo-time, you try to average the gene expression, the average to obtain the average the trajectory, and then you can look off the central trend or the aggregate trend of the gene expression change over time. But in RNA velocity, you are looking, or if you're using vector field, you are trying to learn the trajectory for each individual cells over time. So, I mean, in theoretically, this is the better way to do it, but it may be easier and more efficient to just look to identify genes that change along a central trajectory. But other than that, I think if you can, you should use RNA velocity instead of pseudo-time.
And there are a few bioinformatic tools that have been developed, like Grand Slam and Slam Dunk, specifically to quantify old RNA in metabolic labeling experiments. Could those tools be integrated into your framework, or does Dynamo perform that kind of quantification on its own?
I guess Grand Slam and Slam Dunk definitely can be incorporated in our tool. Right now, Dynamo doesn't have its own tool yet. There are also, but there are a few caveats for just existing tool. So the Grand Slam and the Slam Dunk, if I remember it correctly, those tools actually tries to model the mutation for each individual reads. And if we adapt to single cell, we may need to model this for each individual reads for each individual gene in each individual cell. So this will be, and this, by the way, this is actually what the next SIG paper was doing. So this is this will actually be very time consuming. So in the scale fed and the SCNT SIG method, they are trained to use the UMA and also other simplifications, like just model universal mutation rare, background mutation rare across all the cells. I think we need this kind of simplification to make this mutation estimation more accurate for the huge single-cell RNA-seq data sets.
All right, great. And that's all the time we have for today. So we'd like to thank Dr. Xiaojie Qiu and our sponsor, Lexogen. If we didn't have time to get to your question, we will try to follow up with our experts. As a reminder, please look out for the pop-up survey after you log out to provide your feedback. If you missed any part of this webinar or wish to listen to it again, a link to an archived version will be emailed to all attendees. Thank you for joining us for this genome webinar.
Thank you.